Very brief PowerPoint presentation on backgrounds. It's just to confirm some information about when you are looking at your slide, how you report your findings. We've got that big chart that we go through in regard to infection, inflammatory, neoplastic, non-neoplastic, non-inflammatory, etc. So the other part of it, so of course when you get a sample in front of you, you need to look at the types of cells that you're seeing, classify your sample appropriately into one of those categories, subclassify appropriately, as the other chart goes into in one of your previous PowerPoints. And then the last step as well is to comment on the background. So what are you seeing in the background? Are you seeing lots of red blood cells? Are you seeing sort of a proteinaceous background? Are you seeing heaps of granules? Because all that information gives the diagnostic process more information to work with. So identify your sample, the cells in your sample, note the numbers, and then classify it. So report it in regard to inflammatory, neoplastic, non-neoplastic, non-inflammatory, or infection as well. Identify the background of the sample. So the types of backgrounds that we'll focus on are proteinaceous, granules or granular, blood being red blood cells, and broken cells, all of which can be diagnostically important or significant, and some are less than others, and we'll talk about that in regard to broken cells in particular. Proteinaceous, we've looked at proteinaceous samples probably at this point in the class. I know that we did look at one line smear already. I think it's sample C85, and it's an inflammatory sample. In the background, line smears are really nice to identify proteinaceous samples because your whole sample is sitting on a line, more or less. So you're able to identify, is that background proteinaceous or not, by skipping from the line of cells and back to an area where there's nothing. And that skip between the line of cells and back to the area where there's nothing will help show you, is it a clear background or is it lightly proteinaceous? So is it a clear background or is there a light coloring throughout the entire background? The example on this slide is pretty obvious that it's not a clear background. In particular, you can see little areas of white, little bubbles, and that's just where the sample has separated from the slide. Sort of like when you're preparing a blood smear, and especially if it's a very liquid or anemic sample, then your blood tends to separate a little bit and create what looks like bubbles, but it's just absence of it sticking to the slide. So when you have a slide in general with a colored background, so sort of like a frosted glass background where it's one color, semi-opaque, and it has various levels of transparency, that's called proteinaceous. And there are lots of different reasons for proteinaceous backgrounds, one of which is mucin. So common in joint and salivary reproductive tract to have a mucinous background. Osteoid, dense pink bony material, and then amyloid, meaning protein in general. I'm completely okay if you just describe it as proteinaceous or non-proteinaceous. That's completely okay. I don't expect you to go into great detail about it. Cytologists definitely will look at that proteinaceous background and identify the components within the proteinaceous background and then classify it accordingly. But for our purposes, just identifying it as having a proteinaceous background is helpful. Likewise, the next one would be granular. So granules or granular background, really common with ruptured mast cells and really common with ruptured melanocytes. Now, they can have a granular and proteinaceous background, not terribly uncommon. I have a number of slides that exhibit that. So with that, you'll see, again, a colored background with varying levels of transparency, and then granules throughout. Really important to confirm that those granules are not bacteria, as with anything and with any mast cell tumor, to ensure that it's not bacteria. If it's bacteria, ideally you should be able to focus on each individual cocci or rod, and they should be stained dark purpley blue and similar in their shape and size throughout your sample. Otherwise, granular is acceptable. Okay, here's an example of a granular background. Now this one is more than just a granular background. One of our next classifications is red blood cells in the background. So this would actually be a granular and red blood cell background as well. And you can see these are melanocytes, the two big guys, a little bit strange shape, nucleus to one side, lots of melanin covering. Okay, big melanocyte, little melanocyte. And then when they're disturbed or when they're disrupted in general, when you're poking them with a fine needle aspirate, then they start to degranulate, or they have the potential to degranulate. So they can release some of the melanin granules into the background, onto other cells sometimes as well. But in this case, we created a granular background and red blood cell background. So the next one is red blood cell background. Can cause the presence of red blood cells can cause white blood cells to emerge as well, simply by their presence. That being said, in order to classify it as an inflammatory sample, just look at all the cells that are present. Okay, if you're just getting one neutrophil kicking around and you have a lot of red blood cells, you may have struck peripheral blood. Okay, you may be looking at more of a peripheral blood sample. So you need to see what other cells are kicking around. Do you have degenerate neutrophils? Do you have lots of neutrophils? Do you have neutrophils and macrophages kicking around? All of those would indicate more toward an inflammatory reaction. Just keep that in mind as you're looking through samples. It does get tricky until you've got a pretty good knack for it, but always look at the entirety of the cells that are present and what they're telling you. So is it evidence of a hemorrhage? Quantify with mild, moderate, or marked terminology. We're not diagnosing a hemorrhage, just keep that in mind. But if we're seeing an extreme number of red blood cells, we definitely should note that in our report so that the veterinarian can assess whether or not that is something of importance in regard to the diagnostic process. And then broken cells are also really important to note. I know that you have learned, I believe, to report them on your hematology, so on peripheral blood smear preparation. When you're looking at them, you typically report basket and smudge cells. Same with cytology, really important to note naked nuclei and broken cells in general. It can be caused during collection. That's probably the number one situation that you're going to see broken cells and naked nuclei. But there are certain populations of cells where it's very, very, very common for them to happen regardless of the sensitivity of collection. So I have lymph nodes on here, yes, but thyroid gland cells, so those glandular cells associated with the thyroid, they commonly erupt when they're taken out of the body. And they're very, very sensitive. They're very, very fragile. So to note that you have an abundance of naked nuclei, so nuclei without cytoplasmic borders, is really important because if we're looking at thyroid cells in particular, or we're hoping to look at thyroid cells, the presence of those naked nuclei themselves can be helpful in identifying that, yes, you were taking the sample in the correct area. No, you don't necessarily have a diagnostic sample. But yes, it's very likely that you were in fact taking thyroid cells. Fragile populations as well, lymph nodes are very fragile. So with the collection process, having very light touch, not jabbing around too much as you're going in, can be really important and helpful in getting full intact cells through your withdrawals. So always note if there are broken cells in the background. So as you can imagine, if you have a background where there are lots of broken cells, there are red blood cells, there's proteinaceous background and granules, you do have to note all four categories as being present because it helps with the overall diagnostic process.